Hello, good evening, everyone. Thank you for staying so long for the talks. For my study, I'll just be presenting on some exploratory analysis, looking at how canopy attributes affect bed sediments. So compared to Bernadette's talk, who gave you a lot of information, a lot of data, mine will be relatively short and sweet, and hope it can tide us through the evening. So here we go. Okay, I think seagrass meadows are awesome because their presence actually helps to modify their own environment and in a way facilitate their own survival and stability. So some of the processes that seagrass canopies can effect include sediment, reducing sediment erosion, increasing sediment trapping, and improving light penetration through the canopy. And all these are critical processes that are crucial for seagrass stability and growth. Still, yeah. Point. Okay. So some of the mechanisms that have been proposed and shown includes the seagrass canopy having leaves to reduce drag forces over bed sediments, to reduce resuspension, and seagrasses having roots and rhizomes, a complex system to help bind and hold down sediments. And this has been done in previous studies that look at how the canopy reduces drag forces in wave or unidirectional flow chambers or flumes. And there have been other studies done in the field to monitor and quantify the movement and composition of sediments in natural beds. Most of these studies have been done in monospecific stands, but in seagrass meadows that I grew up looking at, most of them are multi-specific. So that led me to ask this question, like in multi-specific meadows that we find around here, especially in Singapore, where I'm from, I would like to explore if there is a relationship between canopy properties with the sediment size composition. Okay, so we have this project that allowed us to go around meadows in Singapore and some other a couple of meadows overseas to sort of to look at, do some seagrass surveys, collect some sediments. And across the sites, we have a wide and healthy range of seagrass cover. So going to our method, so at each site that we surveyed, sediment cores were collected, and the particle sizes were sorted with a Melvin analyzer. And at each meadow that we visited, we measured seagrass canopy attributes. So how we did that was at each site, depending on how big or small the sites are, we'll just haphazardly throw quadrats of 50 by 50 centimeter quadrats. And within each quadrat, we'll measure shoot density, biomass, the size of the leaves, the size of the shoots, and how much leaf area cover there is. And after that, using ordination, we try to compare how the structure in seagrass canopy and the underlying structure in sediment size composition and how do they relate to each other. So for the sites, so we surveyed a wide area, quite a number of sites, but from all that quadrats, all the surveys that we done, my analysis was just conducted on a smaller subset where within the quadrat, the seagrass cover was relatively uniform in the sense that it's not a patchy meadow that I am going to analyze. So first going to our results, particle size analysis. Okay, let's see the pointer works or not. So it seems that for some of the meadows like Cyrene, Cistus, Tanah Merah, the particle sizes of the bed sediments do not differ greatly between our visits during the 2 monsoon seasons. And after transforming this data, you threw it inside a PCA and try to see if there's some underlying structure in sediment size composition. So a quick, easy way of reading this graph is these are all the sites that are color-coded along the principal component and the first axis, basically, the horizontal one. Sites that are grouping closer to the right of the axis tend to have a higher proportion of silt in their sediments. And sites that group towards the left axis, the negative part of the axis, tend to have a greater proportion of coarser sediments. And for the y-axis, which explain about 30% of the variance, going positively correlated would be, sites which have a higher proportion of sand, which is between the size class of 250 to 500 microns. Yeah, sites tend to have a higher proportion of that size class. And then, so that's it with sediment sizes. And then going on to our canopy attributes, I'll just quickly go through like how all these are measured and how these are derived. So shoot density is basically, I'm just accounting for whatever is sticking out of the ground. So I'm not distinguishing between whether it's a Halophila shoot or it's a Thalassia hemphysii shoot. Just as long as there's one point sticking out of the ground, I count it as a shoot. And then canopy height, which is the average height of all the leaves within the quadrat that we counted. And below, for above and below ground biomass, which is quite self-explanatory, we do see a big variation amongst the sites that we surveyed. So for Labrador, which some of you may be going for the field trip, it's dominated by the very big size Thalassia hemphysii leaves that contributed heavily to the dominance in above ground biomass. While sites in Cistus, it's dominated by the very, very tiny size Halophila ovalis. So if you imagine my pinky finger, the leaves are half the size of my pinky finger, so which makes sorting very painful. And then below ground bio, and also because the leaves are so small, the below ground biomass is also very tiny. So Cistus has consistently ranked as a very low leaf size, low leaf biomass site. And then looking at shoot size, so again, Labrador and Serenat, which are both dominated by the big size Thalassia hemphysii leaves, they have very high shoot size. So what shoot size means is the total amount of leaf area per shoot. So it accounts for the number of leaves per shoot and also the size of each leaf. While in comparison, leaf area index takes into account shoot density. So it's actually a measure of how much leaf area is found within a unit area surveyed. So some of the sharp-eye audience here might realize that there might be some collinearity between these two, which is true. And that's why when I did a linear modeling, when I was trying to find out whether PC1 from my sediment size analysis, how does it vary with all these canopy attributes that I've measured, I have to take out shoot size because it was highly collinear with leaf area index. So what I found from the linear modeling was that below ground biomass was negatively varied negatively with PC1, while leaf area index varied positively with PC1. For PC2, take out shoot size. Oh, oops. And what I found was that canopy height varies positively with PC2, while leaf area varied negatively with PC2. So to put this so that we can see this visually, so I've imposed that, I've ordinated the canopy attributes onto our sediment size graph, and pick out the colors so we can see. So a bit disappointingly is that the blue arrows are all pretty tiny, meaning that correlation is not that high, which is very sad. But never mind, I'll still talk about this. So I'm not sure, okay, so if you remember, PC1, leaf area index, varies positively, so you can roughly say that sites with a high leaf area index tends to be found in areas with greater proportion of silt. While on the other end, amongst that jumble of letters, there's actually a below ground biomass factor there. So sites that tend to have more below ground biomass, it tends to have a higher portion of coarse sand. So if you apply that same reasoning to PC2, positively correlated with PC2 is a higher canopy height and a lower leaf area index. Okay, so that's probably what I said, and I'm running out of time. So it seems that our results was kind of intuitive in the sense that sites with bigger leaf species like your Thalassia and your Cyanamydosia rotundata, it seems to accumulate a greater proportion of finer sediments, while sites with species that have a lower above to below ground biomass, they tend to accumulate slightly more coarse sediments. But really a caveat to this study is that it's just an exploratory analysis, and our interpretation is still subjected to what's the sediment, local sediment availability and transport, or the hydrodynamic processes that we did not take into account before. So what goes on after this is that I'm going to look at using flume and erosion chambers to test out how the different canopy attributes actually perform under various calibrated shear and drag forces, and also take into account, start measuring biomechanical properties of the plants, and also think about plant arrangement, how different plants orientate themselves against a dominant wave water motion. And okay, so my interest in this study is primarily motivated by trying to improve our predictive capability of what happens when we have to remove a whole patch of vegetation, what would happen to the sediment transport and sediment fade of whatever is in that area. So seagrasses not only provide physical resistance to water flow, they also, between bare substrate and presence of vegetation, we probably can expect some differences in microbial communities, and these microbial communities can secrete some extracellular matrix that might or might not help to hold down sediments, we don't know. And also, presence of in-fauna structures. Animals love seagrass, algae love seagrass. When they hide in there, they also present some way to affect flow of water over the bed. So yes, so I'm ending the talk here. Okay, thanks. Okay, sorry, bye.